What happens during a heart attack, and this is just a sketch of a heart, is that you've got an artery coming off the aorta here to supply blood to the muscle. That artery is blocked, and as a result of that, you end up with a heart attack. This area in here, or dead muscle. That dead muscle, initially the heart can compensate for that, but as time goes by, that dead muscle can lead to heart failure. Around the heart attack zone, you've got an area that is potentially salvageable. So that muscle is not dead, that muscle may die, or we may be able to reverse the process. As you can see, if we can limit the size of this heart attack, then the heart muscle overall is going to be better off. And that's one of the things that has interested us from the beginning with our research. And this is research that's been conducted in collaboration between ourselves, the Clinical School at Waikato Hospital, and ag researcher Okura, the Department of Growth and Physiology. So one of the things that we've been looking at is around the border of the heart attack, and what we've seen is that insulin-like growth factor 1 expression is increased. Now, insulin-like growth factor 1 can protect against programmed cell death, or what's called apoptosis, and it also can increase proliferation of muscle cells. So this was of interest to us to see if we could if this was acting as a signal to try and protect the heart muscle. We give the growth factor by two routes. One was subcutaneously, and the other was by an injection into the lining of the heart, the sac around the heart. So the subcutaneous injection didn't improve heart muscle function, but the delivery into the heart sac did improve heart muscle function. We then looked at a variant of IGF-1, a splice variant of IGF-1 called mechanogrowth factor. At the time of the heart attack, we delivered the MGF into the artery that supplied the heart attack muscle. So that's how we delivered the MGF. And what we found with that is that we did indeed limit the heart attack size and improved heart muscle function. So we're currently doing work looking at myostatin knockout, so the absence of myostatin in mice, creating heart attacks in mice, to see if we can see if there's any difference in the heart attack size, and particularly this area around the heart attack that we may be able to influence favorably and prevent the heart muscle eventually failing.